Hello, today I'll give you an update on mapping dependencies in desmoid tumors and developing a physiologically relevant platform to validate putative dependencies. The goal of the cancer dependency map is to understand the relationship between the molecular features of cancer and the dependencies they cause. The aim is to discover targets and drug repurposing hypotheses and biomarkers that predict such dependencies. We learned these relationships by systematically identifying genetic dependencies and drug sensitivities in hundreds of experiments and developing predictive models that take the molecular features as covariates. From this work, we've come to appreciate that gene expression holds the greatest predictive power, and we continue to expand and improve the resolution of this dependency map by screening hundreds of new cell lines. However, we are also learning that cell line generation for the purpose of profiling and validating dependencies has its limitations. The success rate of generating a long-term cell line for many diseases remains low with traditional approaches. We also know that prolonged culturing induces genomic and transcriptional evolution of the cells, and the production of homogeneous cell lines may mask some of the potential effects of cell-cell interactions. Specifically for desmoids, our experience attempting to produce new cell lines or credential existing ones has revealed that the heterogeneity in their composition presents a challenge for cell line generation. We've studied over 40 early cultures produced by various teams and found that over 70% exhibit loss of tumor purity with subsequent passaging. We have successfully produced 2 long-term cell lines with confirmed beta-catenin mutations at high purity that are now in CRISPR and drug screening. Still, this represents less than 5% of samples attempted, clearly supporting the need for a different strategy. So we are asking 2 research questions. First, how can we improve the success of preserving the tumor population in vitro models of desmoid tumors? And secondly, considering the heterogeneity is a feature of desmoids, how can we conduct perturbational studies and map dependencies in early heterogeneous samples without an intermediate culturing step? To address the first challenge, last year we presented a proof of concept for a multiplex bar-coded PCR approach to track beta-catenin mutations across many conditions. Our goal is to use this method to analyze many culture conditions and identify those that preserve the tumor fraction. We have since applied this method on early desmoid tumor cultures and confirmed its reproducibility. We tracked the growth and monitored allelic fractions across multiple conditions and identified those where tumor purity is preserved. While we're still gathering data from additional biological replicates, we are observing trends for some media conditions that show a tendency towards increased or decreased mutant beta-catenin allelic fraction. Our data also show that it is possible to study the effect of different conditions, for example, drug treatment in the future, in a tumor-specific and high-throughput manner. For the second aim, to map dependencies in early mixed samples of desmoid tumors, while deep sequencing offers a solution for quantifying changes in the tumor population, molecular profiling remains a challenge in heterogeneous samples, especially for understanding the gene expression profile. Naturally, RNA sequencing at the single-cell level can offer a solution, but a challenge remains in distinguishing the tumor from non-tumor populations in a disease like desmoids, where both the tumor and the stromal cells share a mesenchymal profile, and the relatively quiet genomics precludes, for example, CNV inference to identify the tumor cells in RNA sequencing data. We therefore asked whether simultaneous capture of the identifying feature in desmoids, namely a beta-catenin mutation, would help us label cells in single-cell RNA sequencing experiments as either a tumor cell or a stromal cell. And we combine this knowledge with the gene expression profile so that we can produce an independent profile for each tumor cell population. So in the past year, we worked on modifying the single-cell RNA sequencing protocol to capture both expression and genotype information. The method involved modifying mRNA capture beads to not only target the 3' end of the transcripts, but also the beta-catenin hotspot region. Single cells are then encapsulated with these modified capture beads in nanowell arrays so that each single cell is uniquely bar-coded. This allows us to run parallel sequencing experiments for gene expression and genotyping while sharing the same unique cell barcode to converge the data in the end. After we perform validation experiments to ensure the modification does indeed capture the beta-catenin hotspot region, we went ahead with a preliminary experiment using an admixture of 3 samples, one carrying beta-catenin T41A mutation, another with an S45F mutation, and a third sample consisting of beta-catenin wild-type cells, all mixed together as one sample, and we used our method to try and deconvolute these cells. Of course, since beta-catenin mutations are heterozygous and given the typically low depth of sequencing in single-cell experiments, it's impossible to be 100% certain that a cell is not a beta-catenin mutant cell if we do not see any mutant alleles assigned to its unique barcode. We therefore defined a 95% confidence interval using cells that we could label as mutant, and cells that didn't cross the threshold are labeled as ambiguous. From this small-scale preliminary study, we were able to annotate about 60% of the cells with beta-catenin mutation status, supporting the feasibility of this approach. And our data suggests that we can reach over 85% annotation success with deeper sequencing. Importantly, we can now study single-cell gene expression data with knowledge of beta-catenin mutation status in each cell. From this early study, using non-ambiguous cells only, we identified 3 transcriptional clusters via graph-based community detection. And interestingly, we find that in this study, the cells we labeled as beta-catenin wild-type exhibited a different transcriptional composition than the T41A or the S45F cells. And because we now have beta-catenin mutation status for each cell, we can perform differential gene expression analysis between the 2 subgroups. Despite the sparsity of this early experiment, we successfully identified several genes whose expression is significantly different between the wild-type and mutant cells, as validated with bulk RNA sequencing. We anticipate that with increased scale and sequencing depth, that we can improve the detection power in heterogeneous samples. In summary, our work focuses on enabling dependency mapping in early samples, which typically present as heterogeneous composition of tumor and stromal fibroblastic populations. To be successful, we need a readout that would allow us to study the effect of a perturbation in a tumor-specific and high-throughput manner. Here, we are developing and applying our multiplex bar-coded PCR approach. At the same time, we also require the ability to molecularly characterize the samples in a subpopulation-specific manner. Here, we modified single-cell RNA sequencing protocols to simultaneously capture both the transcriptome as well as the genotype of individual cells so that we can annotate cell identities and establish a desmoid tumor single-cell reference. We anticipate that the application of both methods will produce a more physiologically relevant target validation platform for putative dependencies that we anticipate identifying from large-scale CRISPR and drug screens using the long-term cell lines we have expanded. Finally, I'd like to thank everyone that has helped with this project and our funding sources. Thank you for listening.